And welcome to Photosynthesis 2030 webinar series funded by the Capitalize project to promote collaboration amongst the photosynthesis community. I'm Dr. Louisa Diva. So I'm delighted today to welcome our two presenters, Professor Roberta Croce and Dr. Sam Taylor, both of which are going to talk to us about how plants deal with problems with light, I suppose, excess or not enough light, shading, and how this affects efficiency of photosynthesis and how their work will target this. I'm going to introduce to our second speaker is Dr. Sam Taylor from the Lancaster University. He's based in the Environment Centre at Lancaster University. He's a member of the RIPE team, and his work focuses on RUBISCO activity and how its activity affects photosynthetic regulation during and following shade in crop canopies. Today he's going to talk about diurnal RUBISCO activity and photosynthesis of induction. Okay, thank you.
Okay, you have my screen?
Yeah.
Yeah, good. Yeah, so I think the important thing here is to realize that everything I say today is about trying to understand RUBISCO. So the group here is led by Elizabeth Carmer-Silva, and a lot of what we're interested in is how RUBISCO is regulated. And my role in that setting, particularly the project I'm talking about today, which is part of the Realizing Increased Photosynthetic Efficiency project, RIPE, is to try and phenotype what's going on with RUBISCO in shade-sun transitions. So just quickly click. My talk today is going to include a brief word about COWPEA at the start. That's because at the end of the talk, in section 3, I'm going to be talking about non-steady state RUBISCO activity in COWPEA. So I need to introduce why you should be interested in COWPEA. In between those two, I'm going to sandwich in a discussion of how some of our ideas about measuring photosynthetic induction and the RUBISCO component of that have sort of evolved over the last few years, and what that means for the methods that we use, because that's important to that final story that I'll tell. So to briefly introduce COWPEA, the reason that we work on COWPEA is it's a really important grain crop in sub-Saharan Africa. So to put that in context, Nigeria, which is predicted to have a larger population than the United States by the middle of this century, produced about 46% of the global dry COWPEA yield of 7.4 megatons in 2017. So we've got a situation where populations that are developing very rapidly have a preference for this foodstuff. It's nutritionally important for them, and there's a real need to try and develop these crops in a way that will help sustain that population growth. So nutritionally important for protein content as a legume, also good for vitamins and minerals. And the way that it's grown in terms of its agronomy is it's often produced in intercrops with maize and sorghum, but it can also be used as a really short duration pulse. So there are some varieties that have a very quick turnaround of about 60 days. And in those cases, those can actually be used as a chaser to a more intensive crop of maize or sorghum in situations where you have a wet and the dry season to kind of get the last out of the soil moisture that's available in farmers' fields. The two illustrations here on the right-hand side are just some of our contacts at Ahmadu Bello University in Nigeria, just to show you what a plot of COWPEA looks like in its native environment. And then also there's a shot here of some of the various varieties we have seed of in Lancaster. We have a large collection because ultimately we're going for a genetic screening of these varieties. And you can see that the standard version that we would see in the shops, at least in the UK, is this kind of black-eyed pea type of COWPEA, but they come in quite a number of different varieties, and that's often something that breeders are interested in when they're looking at quality for their consumers. So having done that, I'll start talking about quantifying non-steady state RUBISCO activity. The interest in this is that photosynthetic induction is something that's happening daily and many times a day for leaves. So we have to deal with the transition from nighttime to daytime. So photosynthesis has to be the various processes in photosynthesis, of which MPQ is one, have to be coordinated to help the chloroplast work efficiently. In the context of, say, a field crop of soybean, if you make a ray tracing model of what's going on in leaves towards the top of the canopy, you get something that looks like this. So this is for a field in Illinois. You have a daily progression of sunlight from low levels at night, none at nighttime, to maximal intensity in the middle of the day. But this is interrupted by a series of periods of shade where you've got leaves that are sitting between this particular chloroplast that's being modeled here and the solar beam. And so you can have quite long periods of shade, short periods of higher radiance, all mixed in together. And the shade, the main thing about this shade is that this is really intense. So this is something like a 10% level of light here compared to these high values in the middle of the day. So we're interested in how photosynthesis copes with that, and a model that's really important to understanding that as a gas exchange physiologist is the photosynthetic CO2 response. This I'm going to use to sort of contextualize induction in a little bit, but just to give a kind of brief overview, if we look at the rate of CO2 assimilation and the CO2 concentration internal to the leaf, then we can model the idea that there is a limitation forced by carboxylation, which would be controlled by VC max, and a limitation forced by regeneration of components needed for the Calvin cycle, which we can illustrate as electron transport. And then we have maybe an operating point for photosynthesis that would be constrained by these upper limits to photosynthesis. Now, importantly, that operating point is a result of drawdown of CO2 from the outside of the leaf to the inside. So there's a resistance of the leaf to CO2 movement, and that means that CO2 inside the leaf is depleted. And those components kind of describe for us RUBISCO's effect, the effect of electron transport components, and then this can be modeled for what stomata are doing to this scenario. When we think about induction in those terms, well, we'll start by thinking about induction. So this is CO2 assimilation, and this is the time since a period of shade. We've got low photosynthesis coming to the end of shade, and then a gradual increase in photosynthesis coming out of shade up to some asymptotic value. So physiologists conceptualize this as being that there's a period here where there's assimilation that's going missing because the biochemistry that we need to do high rates of photosynthesis is still engaging throughout this process. And work from several decades ago now has shown that there are, in terms of the biochemistry of carbon assimilation, there is a fast phase here, which is predominantly driven by IUPP regeneration, and then a slow phase as we go over the rest of this curve, which is linked with RUBISCO activation. We can do the same thing on an ACI plot. So on the right-hand side here, we've got the assimilation rate against that internal CO2 concentration that I just showed on those previous diagrams. And when we plot the induction onto this surface, we can see that the fast phase here is linked with essentially a movement along that dashed line that indicated the influence of stomatal conductance. So here, this is almost a pure biochemical effect with the level of assimilation being limited by what's happening to the slope of the overall ACI response. But as we go along through this induction, this thing will have a trajectory that's ultimately determined by other factors as well. So there's curvature at the top of this trajectory that's linked with the stomata opening during the induction. And so you get this kind of idea that you have different trajectories on this plane based on whether there's a large impact of diffusive limitation or a biochemical limitation during induction. Right, and so you can take that model, and if you can assume that you know what the biochemical component is, you can partition this into something where you say there's a biochemical component here and it's RUBISCO, and RUBISCO is driving things, and contributing X amount of limitation to this. Similarly, we can partition a limitation due to stomatal conductance, but that relies on you assuming what the limitations are. A way to get around having to assume those limitations is to actually try and measure what's going on in terms of the ACI response. So if we can measure at different CO2 concentrations at different time snaps during induction, then we can construct the whole of the ACI response, and we can start to say whether or not what VC max is at different time points, the green line on that diagram that I showed, and what J max is. And so we can get a time series for VC max or J on the basis of that. That's really onerous in terms of doing an experiment. It takes a long time to build up all of the different reps of induction that you have to do in order to get each of those individual points. But we have done it, and we did it with a series of 3 different brassica species. We were interested in these for reasons of the RUBISCO zone being a bit different between them. But when I did the physiology, we also noticed something interesting about how their VC max dynamic fits with their induction process. So what you're seeing on this figure is that VC max along the y-axis, so that's the amount of RUBISCO activity essentially, for Brassica oleracea increases and then plateaus. And in all cases, we've got this kind of asymptotic shape to these curves. But for Brassica oleracea, that intersects with the point where 90% of the assimilation rate that you would expect under steady state conditions has occurred quite late. So essentially VC max change has already occurred before you get to the point where you're saturated for photosynthesis. For these other 2 varieties, VC max change was ongoing in the background when we were reaching a point where we were getting saturated photosynthesis or close to it. So that implies that the limitation states here are not necessarily all about RUBISCO during induction. And if you look at the ACI responses drawn as I drew that first set of diagrams, you can kind of get a sense for how that is happening. So here we've got our operating points, these gray points. And for Brassica oleracea, in the steady state, it's really strongly limited by RUBISCO activity. It's strongly within this zone where carboxylation activity is going to be keeping things limited. For these other 2 lines here on the right, we're coming more into an area where there's an influence of electron transport on what the maximal rate may be. Bearing in mind that there's some error around these points, at least some individuals, leaves and individuals of Brassica napus and Brassica rapa are probably going to be electron transport limited at the point where they're at this steady state. So that sort of partitions these in terms of a species that has really strong influence of RUBISCO throughout induction, and some where that influence of VC max on induction might be of limited duration. That's basically restated here. We can either have persistent VC max effects, and that in terms of when it might happen might be when you have a relatively low steady state VC max, or you have low levels of leaf conductance that are resulting in a strong drawdown of CO2. Or you can have a limited effect of VC max linked with maybe high leaf conductances and maybe really high VC max, so that your limitation is primarily by electron transport and IUBP regeneration. So that means that when we do induction analysis in terms of VC max, we need to do some quality control. We can't just assume VC max limitation dominates through a slow phase of induction, which is where we went with our recent analysis of RUBISCO activity in COWPEA. So to get from some induction time series of gas exchange to something where we're reasonably confident that we can model VC max, we need to pair our induction time series of gas exchange with a steady state ACI response. The steady state ACI response gives us options on getting some handle on what mesophyll conductance and day respiration look like. Those can be channeled into 1-point estimates of VC max, but we need to constrain the period over which we're interested in VC max to that where we think that VC max is actually the limiting thing. And so we do that based on using the maximal rate of electron transport limited photosynthesis and seeing where that's likely to intersect with the trace for actual measured CO2 assimilation rate. Essentially, once we intersect with this, then we're not so sure that RUBISCO is limiting anymore, and so VC max may not be the primary limiting factor. So we end up with this curtailed data set, and that has some issues in terms of this method for trying to understand the parameterization of how RUBISCO is responding to shade. First of all, we've got that complex quality control process. That is then leading to the fact we're having to extrapolate some of our parameters. So if we have an equation here which describes how VC max is changing after shade, these parameters, so the initial and final state, are now extrapolated because we can't actually get them from the data that we know is VC max limited. In addition, if we want to know what's happening in terms of a half-time and a characterization of the decrease of VC max in shade, if this is the only data that we've got, we have to make a series of rearrangements of an equation that describes that decrease, and then we have to make an assumption about how accurate our measure of VC max is in order to get some sort of estimate of an upper limit on a half-time here. So this is all a bit shaky, and particularly the measurement of decreasing VC max is problematic. So to get around that, Emmanuel Escobar built a novel light rig system to quantify activation state in shade. So this basically consists of a set of sunlight simulating LEDs and a water bath. And the water bath lets us keep discs from leaves in steady temperature conditions while we put them through a sequence of light treatments that mimic shade and sun. Being able to put a large number of leaf discs on the system lets us sample rapidly and lets us do many time points throughout an induction sequence, and so we can come up with data that looks something like this. So the benefits of this, we've got rid of the need for complex quality control. I mean, this is not a simple experiment. It's still hard work to do this, but we don't have those extrapolated parameters for the shade component of this anymore, or even for the sun component of this anymore. We don't have to make assumptions to obtain a shade characterization because we now actually have data here that runs through that period. And the whole thing can now be modeled as a single multi-part model, and that's really powerful in terms of being able to use it to make inferences in a nonlinear mixed models context. So when we take these data and we compare them, the light rig versus the gas exchange, we found that the light rig, so the measurements of RUBISCO activation state in leaf discs, showed us much faster deactivation than we could estimate from our gas exchange data set. That's kind of a function of the data set. But also faster activation than we measured from the gas exchange data set. We also got the benefit from the light rig assay of being able to look at differences among genotypes for both activation and deactivation. And interestingly, we found that although activation rates were similar, so the half-time for activation is similar among genotypes here, our genotypes of COWPEA that we looked at were quite different in terms of the rates of deactivation of RUBISCO in shade. Now that's interesting because potentially if you can find something that doesn't deactivate very fast, you don't have as much recovery to do when you do experience short periods of shade. In addition to giving us information about the dynamics of RUBISCO activity, the light rig dataset also let us look at what it was that was causing the deactivation of RUBISCO, or the lower RUBISCO activity. So a RUBISCO activation state assay consists of two components. There's an initial activity and then there's a total activity which accounts for carbamylation by incubating the sample with CO2 and magnesium for a period. Essentially, if your initial activity is changing, that's an indicator of changing carbamylation state. And if your total activity is changing, that's an indication that even when you add CO2 and magnesium to the mix, there is some difference in the amount of available RUBISCO active sites that is probably caused by sugar phosphate inhibition, for example. And what we found was that at least in these experiments, when we go to lower light levels on the y-axis of these figures, the carbamylation state of the initial activity of these leaf discs is lower in that shaded condition, but the total activity isn't really affected. So this is giving us some pointers to look at carbamylation state a bit more in future. And so we're really interested to see what's going on there. So I've whistled through that a little bit quicker than I originally thought I might, but summarizing that, RUBISCO deactivation in these light rig experiments is stabilizing within 2 to 10 minutes of shading. That's quite quick. It's quicker than we really anticipated, and we'd always sort of assumed at least 20 minutes or so. So this opens up some possibilities for what you might do with your methods. You could probably use higher throughput, do more induction measurements because you don't need to use as much shade. The accessions differ in the speed of deactivation, which, as I mentioned, if we have accessions that don't deactivate very fast, they may actually be preferable because they would not lose as much photosynthetic capacity when they go into shade. And finally, we've got this thing about carbamylation rather than sugar phosphate inhibition as the primary driver of decreased activation state and shade. So that's telling us something about the biochemistry that's really interesting, and that gives us some new areas to follow up on. Just to flag these are the publications I mentioned at the start. We're sort of linked together here to put this story together. We've got the one about brassicas, the COWPEA publication in the middle, and then we've just had published this annual review of plant biology where those kinds of diagrams of the induction on the different phase planes are put into context, and we do a bit of review of different measurements people have made over the years. Putting all that together, I also need to thank the team. Not all of these people directly contributed to the work in this talk. Some of them are here because they're members of the team that's working hard at the moment on the next steps in this research. But we're a big team and we have a lot of fun together doing this work. So thank you to everyone in the team. Thank you to those of you who've been listening, and I will happily take any questions.
Okay, thank you very much. That looked like a lot of hard work going on there. Anyone who's used to LICO and have to do all those different measurements, I think appreciates all of that you've done. If there's any questions, please put them in the question box. In the meantime, I will ask a question. I work on Brassica oleracea, and we look at natural variation across the range of morphotypes. So I was quite surprised to see that you could see such differences between the different brassica species, between rapa and oleracea, etc. Given that within, for example, oleracea, the variation is quite large. You know? So
no, I mean, I don't think, in a sense, the species comparison there is a little overblown, right? So we wouldn't necessarily expect that if you went and looked at a large number of genotypes of oleracea, you wouldn't see variation in that. I think I would probably say when we look at brassicas and we look at some different genotypes of brassicas, we can see that there's variation in what's going on in terms of control of induction. And I would expect that if we went to one of those individual crop species and looked at a range of genotypes, we would see some variation. And I think that's important because that's actually what we're doing with COWPEA, right? So in COWPEA, we have a very large diversity panel, and we're looking to see how much variation there is in some of these things. Basically informed by these more detailed assays, trying to use more high throughput phenotyping approaches.
So is the range of deactivation, so you're talking that it was faster than you expected in COWPEA, but what, for example, would be something on the other end of the scale? Is it a really massive range of deactivation that you have measured anywhere?
Well, we were expecting it to be a lot longer, and that's partly because the literature around this was traditionally focused on forest shade species. And so you have these examples from forest shade species where they take a really long time they take 30 minutes or so as a half time for deactivating RUBISCO. So we were expecting that it wouldn't deactivate particularly fast, because that is in itself a disadvantage. If you deactivate quickly, then you're losing your capacity to respond when sunlight reappears. So it is quite striking that this is short compared to some of the things that have been in the literature in the past.
Yeah. I have a question here from Tom van der Berg. Are the different limiting factors to photosynthesis that you investigated in brassica also dynamically changing in the lifetime of the plant? And if variation exists, is it larger or smaller than the difference between species?
I wouldn't be able to put a number on that. No, I mean, I would comment that the way that we do the experiments, we try and keep these things to some sort of comparable stage. These are quite different plants. The brassica rapa is a much faster growing. I mean, it's I think the line that we used is an annual, and the brassica oleracea that we used, I think, is a biennial. And so there's scope for there to be quite a lot of variation in terms of the plant's development in this. But as physiologists, we're interested usually in identifying which phase is likely to be important. For example, in the COWPEA, what's going to be important for grain filling or whatever. To some extent, we focus on stages that we can measure in the lab in convenient circumstances, but there is always that question of which phase during development is important. Yeah, we're certainly not ignoring that.
Okay, I have a question from Christine Rains. To what extent do you think the light-activated enzymes in RUBP regeneration contribute to the rate light induction responses in the leaf?
I mean, the traditional answer is that they're mostly affecting that fast phase and that they activate quite quickly. But depending on the circumstances, they may be coming into play again later in the process. So if there is some slower component there, they may be affecting things. Yeah. I mean, basically that finding from the brassica was to say that the traditional view of RUBP regeneration having an effect only over maybe the first minute or so of induction and then transitioning to everything else being RUBISCO thereafter might be misplaced, because when you look at the leaf as a whole system, you need to consider the possibility that you might be hitting those limits at different times in different contexts as well. So.
Thinking about induction and relaxation speeds, is the one that's better than the other? If you had to choose which one you wanted to improve in terms of crop improvement, would it be the induction or the speed of relaxation there?
Well, the answer in COWPEA seems to be that you would want to look at the relaxation, because we haven't seen as much variation in induction. That said, we've looked at 4 lines in this study, and so the same as the answer about the brassicas, where we looked at 3 representative lines. Once we screen large panels for these traits, then we'll have a better handle on which ones are particularly variable in the panels that are actually bred with. I mean, of course, screening processes are also limited by what the genotypes are that you put into them in the first place. So, yeah, in COWPEA, it looks like deactivation is a good one to go for, because there's evidence of really strong variation in that, but it may be something else in a different crop.
Okay. If there is any more questions, let me know. I've just got some thank yous and nice work. People appreciating your talk. So I will call your questioning to a close then. Thank you very much for a very informative talk there for us. And to let you all know that this was the last webinar of this year before the summer, and we begin again in October, when we have Steve Long going to do the first seminar for us. Can I have the next slide, Christina, please? Steve Long will be talking to us on the 4th of October. So join us again for this. If you're interested in more events that the Capitalise team are running, log on to our website and have a look what's going on there and join the newsletter. And yeah, we'd love to hear from you if you have any comments about the webinar series. Otherwise, till then, goodbye till October. I look forward to you joining us again. Thanks very much. Goodbye.